Okay, good afternoon. Welcome to the 8th colloquium of this season. I'm glad that we are alive, still alive, that we can still be alive, yeah. So our today's speaker is the medical superstar, I would say, medical physicist superstar, Dr Mackie. I will briefly introduce him. So he has a BSc in physics from 1980 from the University of Saskatchewan and a PhD in physics from 1984 from the University of Alberta. He was a professor at the University of Wisconsin from 1987 and currently has an emeritus status. He has been awarded the Coolidge Gold Medal from the AAPM in 2014, the Gold Medal from ASTRO in 2019, and the John Mallard Award for Innovation from the IOMP in 2019, and is a Fellow of the National Academy of Inventors. He is a commissioner of the International Commission of Radiation Units and Measurements, as well as the vice chair of its board. Dr. Mackie has a long entrepreneurial career both as an academic and since then as retiring as a professor. He is best known for tomotherapy, an image-guided intensity modulated radiotherapy company. He is a co-founder of Leo Cancer Care, which is developing upright radiotherapy, on Ljubljana, a fluorescent-guided surgery company, and a board member of the Shine Medical Technologies, which is developing medical isotopes using fusion and fission nuclear reactions. So I'm really glad to have you here, and I invite you to the stage.
Thank you. So I am also on the board of Cosylab, which is, of course, a Slovenian company. And it's great to be here in Slovenia. It's a wonderful country, and it's a really beautiful city. So my talk is on innovation in radiation medicine. And I show you the covers of Time magazine, which are, Time produces an issue on breakthroughs in cancer about every 10 years. And I show you the last three. And in here, you'll find nothing about radiation medicine, nothing. So in 94, it was on cancer genes. 2001, it was on targeted agents and Gleevec. And in 2013, they talked about 800 targeted agents and talking about survival rates improving. Well, I can guarantee you nothing that they described improved cancer rates except early detection because of better imaging. Radiation oncology are really the reasons why the cure rates have improved. This is the official disclosure list. And you won't see on this Cosylab, this is from an older slide, and I failed to put it on. So the contents are, talk a little bit about academic innovation and its relationship to company formation. The fact that radiotherapy has been disrupted over the last 20 years. I will talk about targeted therapies, and there is a relationship then to the Time magazine articles, because they're using the same target, could use radiopharmaceutical therapy, and boron neutron capture therapy, for example, could use that as well. And I'll talk about imaging data science and the opportunities there. So I'd like to talk a little bit about innovation in academia. And this was a study that asked various types of investigators if they were in a spectrum between pure basic to pure applied, with equally basic and applied at the center. And you see that most people in university would say that they're equally basic and applied. Extremities of that are humanities tend to be more theoretical, whereas agriculture and medicine tends to be more pure applied. So we are, and physics and mathematical sciences and medical physics are kind of straddling that spectrum. So we do applied science, and applied sciences should be applied. And I'll be telling you that the only way to get medicine applied is to apply it through a company. Well, let's also look at one of the interesting things about companies is that they're doing most of the research now. In terms of research spend, companies are outpacing governments by at least a factor of 2. And that's not the way it used to be in the Cold War and the Nassau period. Governments were funding most of the science, at least in the United States. So what is the role of academia and industry? And I like the analogy of a mine and a mill. So the university is a mine. They mine ideas. And a company is a mill. They process the ideas and they get it out to market. So a mill won't last long if there's no mine, but a mill is required to take the product to market. And what's most important, I think, in medicine and other highly regulated industries is approvals. And in the United States, it's the FDA, and in Europe, it's getting CE mark. And so the part I show here in red is the part that really is company is required. So the invention and prototyping, preclinical studies, that's all very good in a university, but as soon as you want to get involved in regulatory work and being able to create a medical device file, for example, you need to have a company. Now, one of the nice things about our particular field of radiation medicine is there's lots of predicates. And if you can have a substantial equivalency, then you can escape clinical trials and get it approved in the United States in a so-called 510K. So there's two really types of innovation, sustaining innovation and disruptive innovation. And in sustaining innovation, the problem is well understood. In disruptive, the problem is not well understood. It's more research. There's an existing market for sustaining, and you often have to go into a new market for disruptive. Innovation improves performance, lower cost, and has incremental changes, whereas innovation is dramatic and sometimes game-changing. The customer is believable because they understand the products, but the customer might not even know a problem exists, actually. And so they might not be as helpful. Market's predictable versus unpredictable. And traditional business methods are sufficient, whereas traditional business methods often fail in disruptive technology. And it turns out that universities are best at introducing disruptive innovation because they're really exploring the new frontiers. And they have PhDs that need to do new things to get their PhD. And existing companies can best manage incremental change. And they often have product plans years out in the future, and they don't want to disrupt that. And patents they've applied for future products years out. And disruptive technologies upset the current product lines. And so very disruptive technologies are best commercialized by startup companies, or the largest companies may have isolated research groups that can do disruptive things for them. Medical physics has a particularly interesting role in a university. Imagine that you are a civil engineer and you design bridges, right? Wouldn't it be great if there was a bridge building company embedded in your School of Engineering that you could practice on, give them ideas and get feedback? Well, that's effectively what we have in medical physics. At least in the United States, those are businesses. That's a business operating in the framework of a university. And it's not that much different in Europe. These are businesses that talk about profit and losses just the way businesses outside of hospitals do. By the way, the bridge on the right is not a bridge at all, but a canal to lift boats. So I'm going to spend quite a bit of time talking about the following flow chart that's really important if you want to start a company. You have to identify a problem. You have to develop a solution. You need to protect the IP. You need to understand your customers. Get your customers to understand you as well. And then get investment. So you need to identify an important problem. What's an important problem? Well, it's a problem that causes people lots of pain, even if they don't necessarily know it. And it's an important problem for the business side because it will make enough money or have a big enough market. Arrive at a technical solution to the problem and see if there's IP already around it. There might be patents that have expired and then you have freedom to operate. Or there's an opportunity to get a new patent or copyright. Sometimes for software, a copyright will work. And if you can get a patent or IP protection, then get it. And then the next one is, will sufficient customers like your solution? And if they don't, if you can't find the market, then you need to arrive at a different technical solution. If they don't like your solution, you maybe have to pivot or change or listen to them to arrive at something that there is a sufficient interest in. And then finally, do investors like your business plan? And the pivot there is, well, maybe you haven't identified an important enough problem. So I'm going to go through some examples in my career on sort of answering these questions. And not like I did this retrospectively. I was stumbling ahead like everybody else. But now at least I have some sort of systematic idea how to approach a problem, and I want to communicate that to you. So the first company I was involved with was a company called Geometrics. It was named Geometrics because there's a letter from each of the founders' last names in the word Geometrics. And we had been working on radiotherapy and radiosurgery and radiotherapy planning at the University of Wisconsin. And we had to develop, we wanted a stereotactic radiosurgery program, but there wasn't a 3D treatment planning system, so we had to develop one. We couldn't buy one. And in 91, the FDA told us that medical software would soon need an FDA 510K clearance. The hospital wouldn't fund it. They had funded the software, but they wouldn't fund getting the 510K approval. In fact, they said, well, gee, I guess we can lay off some staff now. So Mark Gehring and Cam Sanders left the university and started a company, and Paul Record and I stayed back, but helped them on the board and as much as we could late at night and on weekends. So let's look through this algorithm then to see what the points were for Geometrics. So there was a need for a 3D CT-based radiation therapy treatment planning company. That was known. People knew about that. And why? Because we were using conventional planar radiographs to plan patients. We really weren't using CT, or very rarely were we using CT back in the 80s. And so it was very specialized cases. And there wasn't any really good 3D treatment planning system on the market. So our idea was to convert our university-backed stereotactic radiosurgery system into a general RTP and get a 510K clearance. So that was our solution to the problem. We had IP protection because we owned the copyright. We had written it. And interesting, the University of Wisconsin, if there's no federal funds, you own your intellectual property at Wisconsin. It's one of the few universities that doesn't claim a work for hire on everything you do. Our only customers were our staff, because we had a treatment planning system for radiosurgery. So we were developing a dose calculation algorithm as part of my research group. So the investor was a company called ADAC, which was really in the nuclear medicine business, and they gave us a very academic advance against royalties, like a book deal. So they would fund, but they would take their funding out of our royalties when we got 510K approval. Well, so we got 510K approval for ADAC to market it, and then they say, well, it's time. What are you going to do? How are you going to fund yourself? Because you owe us $3 million in royalties. And so maybe in 2 years or 18 months, you'll start getting checks. What are you going to do until between now and then? And then they walked away and made us sweat. And then they came back a couple of weeks later and gave us a deal. Tell you what, why don't we give you $4 million and we'll own it. And of course, it was probably worth at least 10 times that. But anyway, it got out into the marketplace. I paid for a house with it, with my shares. So we were happy enough. Eventually, Philips acquired ADAC, and an interesting story there. They bought it for its SPECT scanners, a nuclear medicine business. And it turned out that the only thing profitable about ADAC was Pinnacle. Pinnacle was the only product making money in ADAC. Anyway, it had great success. Hundreds of thousands of patients per year were planned with it. Probably many of you have used it. It was withdrawn from the market last year after 25 years, which isn't bad for software. Software can last for 25 years. That's pretty good. And what we did put into it was a convolution superposition model-based treatment planning system. I won't go through this diagram, but literally there's things in that code that try to model both the electron transport, the scatter well, the head scatter, and electron contamination. So it's not easy to develop a planning system. It also had very good 3D display. It was really the first time in medicine at an excellent, fast 3D display. And so people bought it for the sexiness of it. It was really cool software. But it also had good dose calculation algorithms. And it wasn't the only 3D treatment planning system. There was others. There was a company called CMS from St. Louis. There was some companies in Europe, including one that Varian bought from Finland and made it into Eclipse. So 3D treatment planning displaced simulator equipment. So when I said, in the periods of the 90s, eventually CT will be how we do treatment planning. We won't use simulators anymore. People said, nah, that's too expensive. No, we won't do that. But it did it. It killed it. When was the last time you saw a simulator? Does anybody have a simulator?
I learned at the facility in Preston they still used it in 2021.
Okay. That's fine. But I bet you they didn't buy it new. So it really did disrupt the main planning technology in radiotherapy. And then IMRT was coming along. So at the same time that I was working on Pinnacle, I was working on IMRT, and the idea of tomotherapy started to dawn on us. And this slide from Anders Brahma sort of shows some of the roots of the idea. By the way, if someone ever gets a Nobel Prize for IMRT, which is unlikely, because again, the cancer mafia doesn't think much of radiation therapy, as Time magazine has proven. But if someone were, it would be Anders Brahma. And so he made the following thought experiment and mathematically described it, where imagine that you have blocks that you rotate around the patient, delivering a uniform field. You're not going to get a uniform, if you go across here and measure the dose, you're going to get this kind of sawtooth function. You need to put very high intensity, just missing the organ at risk, but going through the tumor, you need to make this very high. In fact, in theory, infinitely high to get a uniform dose across there. If you do rotation therapy, okay? And then the other very interesting thing is that just as the pure thought experiment, if you imagine, if you had 5 fields, right? You had 5 uniform fields, and then you would see if that was the most optimal, then as compared to some optimal intensity modulated field, wouldn't it be very fluky if it turned out to be uniform? How unlikely would that be, right? It never turns out to be that way. A non-uniform field is always the most optimal. Always the most optimal. So what does that say? That says to me that IMRT is going to kill, conventional conformal therapy, right? Even back in the day. So the tomotherapy academic history was, we had the first ideas about 89, almost the same time as we were developing the treatment planning system. In 91, filed a patent with the Wisconsin Alumni Research Foundation. They're an interesting organization because they're not owned by the university, they're owned by the alumni, right? There's 400,000 living alumni. And so no one owns them, right? It's like the Catholic Church, right? Or the Green Bay Packers football team. So the patent got actually licensed to a company called Nomos, which delivered so-called serial tomotherapy, slice at a time, or 2 slices at a time. And then Wharf gave them the license for an add-on collimator, binary collimator, and was reserving a helical version for somebody else. And in 1984, GE Medical decided they were number 4 in the radiotherapy business, so they said, hey, this might be a way to get us up to number 2. And so they started a research project with us. And what you're seeing there is the GE version of tomotherapy with the GE Discovery bearings and support system with, again, a LINAC and CT scanners on board. But they got out of radiotherapy. They suffered one of those Therac 25 accidents, as I think I mentioned to you, and got out of the field. And then we had to start a company. I had 20 people in my group at the time, so you start a company or you perish. So the startup scenario is, what's the problem to solve? Well, IMRT is more conformal to the tumor volume always. To develop a helical tomotherapy, fan beam tomotherapy system that also does CT, because you're in a CT geometry. So it can provide a way to do scanning of the setup to make sure the patient's set up correctly. We were using a megavoltage beam turned down to a mean energy of about 1 MeV for that. We got IP protection from WRF, and not only did WRF give us IP protection, is in that interval when GE got out of the business and we were forming tomotherapy, they gave my group 1.1 million to keep the efforts going until it could be funded, until we could take their license. The same license that GE had signed was taken away from them because they had the right, WRF had the right to take it away and transfer. So one of the interesting things is we did have a base of customers. All those Nomos customers, Nomos had sold 70 machines. And the people kind of liked, treating two slices at once, because there really wasn't IMRT available. They were the first IMRT company, but they thought it was clunky. So instantly we could make a lot of sales to the old Nomos customers. Of course, it cost a lot more, but it did a lot more. We had a Madison-based, a local investor as our first investor. We also got some money from the state, a loan from the state. So tomotherapy in 2007 was worth more than $1 billion, actually. So it's called a unicorn when we took it public, and merged with Accuray in 2011. But still, about 200,000 patients around the world are treated with tomotherapy every year. So let me just give you an example. Whoops. To give you an example of basically how tomotherapy works. So we were delivering the radiation with slices with binary collimators being able to modulate the beam. So tomotherapy provides the highest modulation of any form of therapy. So this is for total marrow irradiation. You just want to irradiate all the bones and then spare the sensitive tissue. So you can see then that it really can provide extremely conformal dose distributions. And here's a bunch of postage stamps plans put together, so you can see how conformal they are, like just treating the scalp and avoiding the brain, for example, or treating a breast that has a sunken chest, and very complicated mantle fields, that look like the letter A. So IMRT displaced LINACs without MLCs. So again, not just tomotherapy, but other forms of IMRT. So today, nobody buys LINACs without IMRT, right? You probably still have them, but you don't buy a new one without IMRT. So it was disrupted. And then the other thing that tomotherapy had was a CT on board. So we could then compare the simulation that the planning was based on with the image of the day, and then be able to adjust the field. So we were the first company to deliver, if you like, real-time, or not real-time, in-room IGRT with CT. And in fact, now there's lots of image-guided machines besides tomotherapy, ViewRay, Halcyon, the Unity, Reflection, and the Aurora machine from my old alma mater, Edmonton, developing this system. But one thing common about this, these IGRT machines are also coplanar only radiotherapy machines. Okay, so you don't need to have a gantry that has the couch, because rarely do we move the couch anyway. We tend to treat most of our patients without moving the couch in the coplanar position. But now, there's no way that you're going to go buy a LINAC without IGRT. So even if it is one that's a C-arm that allows the couch to move, you're not going to buy it. Okay, they've gone away from the industry. So that's the third disruption. All right, now let me pivot a little bit and talk about proton radiation therapy, because this conference is sort of emphasizing proton radiotherapy. This is the largest gantry in the world in Heidelberg, 800 tons. It's in a hockey rink kind of sized, two-dimensional footprint. They built another vault for a second gantry, but decided not to put it in there. They have robotic couches then in those gantries. But there are systems that do treat patients upright with CT guidance. So this is one that's in the Proton Center in Chicago. There's a robot that places you can put a patient on a chair. The chair can be put on a robot. The robot can move the patient from a CT scanner to the nozzle across the room. It's a little bit clunky though. So a startup scenario for Leo Cancer Care, which I started a couple of years ago with a guy named Stephen Toe, we think there's an opportunity to treat patients in the upright posture with better control of the patient and better control of the posture. And so the solution then is to design an upright positioning system with multiple postures and a CT scanner that can scan in various treatment positions. The IP is protected only by company patents. It was developed outside of university. Customers at first didn't think the patient posture was a problem. Radiation oncologists weren't knocking on the door saying, develop a system for us that would treat upright with a variety of postures. It was something that we couldn't ask customers to really help us with at the beginning. The investors were global. This company was actually started in Australia. It went bankrupt because they couldn't raise capital. And I bought it out of bankruptcy. Why? Because I also had started an upright veterinary CT scanner company that scans the legs of horses 2 at a time and then rotates 90 degrees and will scan their heads and necks. So the first dual direction CT scanner. And that's the sort of scanner that you'll need to treat humans upright, a scanner that can now not go up from the floor, but down from the ceiling. How's it going? Well, you can sell protons without regulatory approval. It's one of the exceptions because they're so expensive. So we have some sales already. The positioner in the CT is being commissioned. There's a photon version under construction in the UK. And we're using Cosylab software services. So to save on staff costs, Cosylab is writing all the control and much of the display, or in fact, all the display software. Well, what about the coplanarity of it? Well, it was shown by Thomas Bortfeld, who I think will be coming here tomorrow, that and this is for a skull-based and C-spine radiotherapy. That even there, where you'd think you'd need a lot more non-coplanar fields, almost all the fields are coplanar. And there's basically peaks in this phase space of couch angle and gantry angle for vertex fields, either at 90 degrees or at 270 degrees. But other than that, most are coplanar. So most patients, and this paper says most patients can be treated with a coplanar machine only, like the Unity or the ViewRay. And so on, and for photons. So what we set out to do then is find a system that controls various postures. So we want to have the patient in a seated position for head and neck, whereas in the prostate, we wanted to be able to have the patient in a semi-standing position, we call it the perched position, and to be able to control these postures so that the patient can't move. And these postures can treat more than 95% of cases. And so the patient position we developed is shown here. It's actually being tested in Lyon, France by Vincent Gregoire at his cancer center, Lyon Bernard. And so the motors above the floor are controlling the patient positioning. And then below the floor, there's 6 degrees of freedom rotations, yaw, pitch, and roll, and translations, X, Y, and Z is being elevation. So so the scan, and of course it needs a scanner that can scan along the angle. On the back, the seat rest can change positions from forward leaning to back leaning. The scanner has to be able to scan in treatment position So it has to be able to scan down there. And the design is basically have the scanner move down arms, which are held at pivot points. And it's fully balanced so that actually a therapist can pull the scanner down and then it scans away from the patient going up. And then if you want to turn this into a CT sim and do conventional patients, you have to move it to 90 degrees, much like we had to do, or in a similar philosophy as we had to do for the horses. And this just shows the patient positioner system. There's some fat fellow here in the picture wearing a mask, so I can't identify him. And so you see what the imaging display was. We freeze an image so that you could compare if the patient's set up correctly. And then this shows some tests of the CT scanner. It's running only half. It's rotating at half speed here. And here you can see it slowly creeping up, scanning. I think this was something like 12 centimeters, and so it would go twice as fast, and then can scan faster. And so this would be then around the patient. The therapist would have pulled it down to the start position, it starts spinning, and then goes up. This is kind of an artist's model of what a room would look like for proton radiation therapy. We call it the Marie system after Marie Curie. And we name all of our products after women who we respect in science and have had less press than they probably should have had. And so the nozzle's coming out, a fixed horizontal beam, a proton beam in this case, would be coming out from the wall. As I said, we're using Cosylab because we think that we can add a bunch of imaging systems more easily to this. It's easier to add imaging systems onto this framework, this platform, than it is on a gantry, right? Because we can have, for example, this is the machine you saw earlier. This is Proton VDA's proton radiography or even CT scanner system that would slide down from the wall and dock into place. And then we're using Cosylab's framework there so that we can add potentially other imaging systems to it, like prompt PET, like maybe a cone beam CT for the X-ray version, where a dual energy fan beam CT scanner might not be needed. And then so they're doing the control as well as the presentation. So this shows the difference in size of a gantry versus an upright system. And in fact, this is the gantry that is going with one of our upright systems to the University of Wisconsin. And so it's, the volume here is, on the order of 20 times less volume that needs to be shielded for this. And of course, the main shielding is only in the direction where the neutrons are going to be going in a similar direction as the protons. So we believe that a gantried machine, there's some uses for a gantried machine, like maybe treating kids that have to be under general anesthetic, leg sarcomas, adults who cannot sit down for whatever reason, and adults needing general anesthesia, perhaps. But we think the vast majority of patients can be treated in a rotating patient instead of rotating gantry framework, so that this will be the workhorse machine and the specialty machine will be the gantry. All right, let me change fields and now talk a little bit about radiopharmaceutical therapy. I think this is going to be a thing, and I think it's going to be a thing that we're going to participate in. So of course, you know that radiotherapy beams go through, provide dose outside of the tumor volume, whereas at least radiopharmaceutical therapy, if tagged with a high-performing agent that only goes to the tumor, you can lower the dose a lot. But there's something that I call the visualization gap that needs to be thought about. If you just naively think about as you increase by decades the tumor cell density, external beam radiation therapy is extremely effective at even very high tumor cell densities, right? We don't need the cells to be labeled. we have rays that are passing through, causing ionization, causing dose to be deposited. Whereas targeted agents are probably only effective at smaller tumor densities. You need to label all the cells. And so there is this visualization gap. Radiotherapy, then, if you can't see the tumor, you can't aim your beam at it. Whereas targeted therapies, you don't have to aim your beam at it. It finds its way to the target. The targeted agent finds the target. And so, so there's two types, there's two classes, if you like, alpha and beta. Alpha have very short range, on the order of cell diameters, whereas beta can travel actually many cell diameters. And you don't have to necessarily label every cell with the beta emitter, but it's not high LET, whereas the alpha is high LET. And in my estimation, a good diagnostic agent should be a good therapeutic agent. So people call this combination either therapeutics or theragnostics. And I think the key is finding a good targeting molecule that you can attach stuff to it. So it's like a truck. It's a truck that you put packages on that's delivered to one address, the tumor. And so we should concentrate on developing robust targeted agents, ideally pan-tumor agents, ones that somehow are differentiated on the basis of neoplasia or not neoplasia. And use the synergies of these tags on targeting agents for the whole life cycle of the tumor. So here's basically the idea. So you have a targeted agent, and even for things like surgery and screening, if you put on a fluorescent molecule on a tag onto the targeting agent, it's useful in screening. It'd be useful for a cell sorter, for example, to be able to find circulating tumor cells. And in surgery, for the surgeon to actually see in real time where the tumor is, even down at the cellular level with microscopy. And then, of course, you can label with MRI, for the MRI, PET, or gamma, so that you're able to then use it for diagnostic radiology and for response assessment. And surgery, by the way, if you label it, it'll still be there for the pathologist to take a look at. And then, of course, we have conventional radiotherapy. But I think that we'll be starting to use radiopharmaceutical therapy as an adjuvant, because we know a lot of tumors like pancreatic tumor and lung tumor, the patient already has metastases. So we will be using these radiopharmaceuticals as prophylactic agents at the time of radiation therapy, right? So it'd be really nice to do the treatment planning of dose so we can take those into account. And then, of course, BNCT. BNCT can also be used. It's really kind of a form of radiation therapy, but has the ability to have these high LET particles in the tumor. And it's coming because now, instead of 1 boron per molecule, people are finding ways to put 10 or 12 borons per molecule. So anytime you increase something by a factor of 10, there's got to be new opportunities. That's the way I look at it. And then, so let's talk a little bit about the imaging systems. So this was some work that we did when I spent 5 years of my life not doing radiation therapy, or radiation medicine, really. So I went to an organization called the Morgridge Institute, which is probably a lot like this institute. It's a multidisciplinary research institute. And one of the things we did was develop a way for microscopists to keep the lights on. Because typically they have to work in the dark, right? They set up the microscope and then turn off the lights and then work in the dark, because the lights will interfere with the fluorescence work that is the workhorse, really, of modern microscopy. This is actually a 2-photon technique that we're looking at here. So A and D is what is with the lights on, but just steady lights on. Of course, the image of A is it's just washed out. You can't see anything. The microscope image completely washes out the fluorescence. It's gone. In the next one, the lights are off, and you're getting a nice image. the next set, the next column. The third column is we call transient lighting. So we're having bursts of light to illuminate the field, but the lights are off, or the lights are only on in the raster pattern when the raster scan goes off the edge. There's a time then that the excitation laser is not producing fluorescence anyway, so you might as well turn it off, right? Or turn on the white lights. So you then have these bursts of white lights that the eye can't tell that it's not steady, right? So you can now go work in the microscopy room while you're acquiring the image. And this is basically how it works. So we use 180 hertz cameras to look at both a white light image and a fluorescent image. And of course, the fluorescent image, the fluorescent camera, we have to have special filters in front of it to get rid of the excitation light as well. So basically then the period's 5.5 milliseconds, with the white light portion being 0.55 milliseconds at 10% duty cycle. And about 90% of the time you can get the fluorescent signal free of any white light, right? And so this we said, well, this can be implemented in surgery as well. Because surgeons now turn out the lights to inspect the field with fluorescence. Well, first get a dark adapted, right? And then inspect the field. If they can operate and then get the cameras to display false color, then they're much happier. So that was the scenario for on-loom surgery. So the problem to solve was fluorescent-guided surgery with the lights on. So imagine that, surgery with the lights on. That's a good problem to solve, right? So the solution is high-speed dual cameras. They're looking down at each other using a lens splitter, a prism to split the beam pass. And even at half the signal, you're throwing away half the fluorescent signal. It's still lots of light, and it's free of contamination. So we had, well, interestingly, we had filed patents at WARF, but we were beat by, happens to be a Nobel Prize winner, Roger Chen at UCSD, who beat us to the patent. It described almost exactly what we were doing. And we said, oh, shit. But then no one had licensed it. Because why? Because it was disruptive. So we went to UCSD and said, can we license it, please? And they said yes. So it's really based on a Nobel Prize winner for green fluorescent protein, GFP. And then a company called Novadeck had introduced fluorescent guidance, but not with white light imaging. It was purely just the fluorescent signal, but not superimposed. So we're going after their customers, and we talked to their customers, and they helped to guide the product. And investors tend to be mainly family offices. There's been a transformation lately. The most money being spent now on startups is rich people's family offices that they invest in stuff, right? And they get professional investors. And a lot of them have reasons, right? They maybe had a family that had cancer. And so it's one of the best places to get medical money these days. So how's it going? Well, we got FDA clearance for vascular surgery because it can also be used with immunocyanin green as a vascular dye. And we're working with research groups on the cancer and nerve targeting. Imagine being able to see nerves, right? Being able to see the note. In fact, imagine that you see in 3 different colors. You see the nerves, you see the vasculature, and you see the tumor. And this is, I won't go into this, but this is, it turns out to be complicated. You're manipulating in real time, 180 hertz, high resolution signals. Turns out you need very special, fast electronics, and so that's really the key. And this just shows, this is a mouse getting a tumor excised. And the circle, you'll see later, there's some green in there as well. So, and you can see then being able to see where little specks of tumor are important. You can just barely see, just make it out just about there now. See that little green dot? And so this then, you can see how easy it is to cut out the tumor, take that green blob away. And there's just a little green blob sitting there. It's hard to see with the lights on here. So let's go back to, I was giving you a bigger picture of a targeted agent. So you can label them with a fluorescent agent, but what about labeling with radioisotopes, as we talked about before? So, there's kind of the usual paradigm is what we're doing today. And the usual paradigm is non-personalized. It's based on activity per unit body mass, right? It's like any other chemotherapy drug. You inject it in. The trouble with that is that intrinsically with the FDA or CE mark approval process with clinical trials, that you have a dose that's limited on the people that are most sensitive to the radiation as a first do no harm, right? So it turns out that some people, more of the radiopharmaceutical product is going to things like the liver, the kidneys, or the bone marrow, and not going to the biodistribution on some people is less good at getting it to the tumor, right? Or the converse, the liver or the bone marrow or the kidney is soaking it up more. And so you're fundamentally limited the dose you can give unless you can personalize, unless you can do dosimetry by injecting a test dye into a patient and then seeing what the biodistribution is and then doing real dosimetry, Monte Carlo-based dosimetry on it, treatment planning for radiopharmaceuticals. And so the ICRU has, with the the chairman of the committee, George Sigoros, this year's ICRU report is about dosimetry-guided radiopharmaceutical. So it's going to bring radiotherapists into it, because we understand planning, and it should also be integrated with conventional external beam planning. I won't go through all the details here, but the ideal radiopharmaceutical paradigm would certainly apply well to beta emitters, which all have the ability to see where those beta emitters are going to, even if it's just a low dose of the therapeutic drug. And then, but it does require imaging. It does, because you need to create a model of the biodistribution, right? That's essential to know to do the dosimetry. So maybe 4 or 5 nuclear medicine or PET studies are required in order to do this. And by the way, there's a whole slew of radiopharmaceuticals. There's only a couple that have been approved today. But there's a whole slew, and they're just being, again, injected dose per mass. So the rest of these, hopefully, or at least some of them, will start to be coming through so that, well, physicians can always, they don't have to follow the guidelines of dose per mass, right? They can do whatever they want. So if they can personalize the dose on the basis of dose guidance without FDA or CE mark. But it'd be nice if the whole process was starting to go in that direction. So I'm on the board of a company called Shine Medical Technologies. In fact, I'm the first investor in the company and the first founding board member, other than the founder. So the problem to solve is I don't know if you know the saga of the supply of Moly-99 for technetium-99m. It's engraved, it's been fragmented for the last 10 years. The Canadian supplier got out. The Canadians built 2 reactors to make Moly-99, and both of them failed to get approved in Canada. So the Canadians gave up. America got all its supply from Canada. Europe's getting its supply from 2 aging 50-year-old reactors in Belgium and the Netherlands, and a little bit coming from South Africa and Australia. So the world supply of Moly has been crippled. So the solution is to build a subcritical assembly fission system to use an aqueous subcritical assembly, which, by the way, was the first nuclear reactor. The first nuclear reactor at Los Alamos was a liquid, a uranyl sulfate reactor. So they're using uranyl sulfate as the fission source. It's nice, but it's aqueous because you can strip off the Moly-99 every weekend and just reuse the uranium. Whereas the conventional supply was using foils of highly enriched uranium, bomb-grade material, actually, that was then just put into waste deposit, not recycled at all. So WRF was the IP protection. So the customers are the reactor-based fission isotopes, which are very unstable. Huge investors, the biggest investors in medical products in the world are these 3 companies, Fidelity, Deerfield, and Oaktree. and they're all in to this company. They've gotten 450 million in investment. The construction's underway on a Moly-99 plant near Madison that can supply all of the U.S. and Canadian demand. Similar plants near Groningen in the Netherlands being planned. They also have been making their own lutetium-177 for therapy. And they even make the deuterium-176 precursor they have an enrichment technology they developed. So SHINE's a pretty cool company. This is the founder, Greg Pfeiffer, a young guy. He's probably about 43 now. This is an older picture. You can't see this now, but this is a 300 keV deuteron source that goes into a tritium gas target at fairly high pressure, but it's on a column. It's on a vacuum column, so you need differential pumping to separate the high gas tritium from relatively high density of gas tritium from obviously being from the accelerator, which needs to be a high vacuum, right? So they managed to have a 6 order of magnitude pressure gradient across that. This is the plant getting constructed. Each of one of these will have one of these systems in it, and this is what it looks like now. It's pretty cool. And as I said, BNCT is coming back. BNCT requires ideally an epithermal neutron source. SHINE could produce some of that, but there are 2 existing companies in the United States that do boron neutron capture. One of them, Neutron Therapeutics in Boston, and one in California called Tri-Alpha Energy, which is also making fusion. They're trying to make fusion as well. They have a BNCT. So it's probably crazy to compete with them, let them have that market. But it's going to be an interesting market. So I'm going to switch gear for the last time and talk a little bit more about quantitative imaging. So we're getting more and more images that really require heavy-duty quantification. But today we're imaging in a qualitative way. So here's a quote. Our imaging reports are almost without exception unstructured, and our medical images are rarely tagged in such a way to be discoverable or useful for data mining efforts. So a report is, for example, in this case, there's an actual report, a mild shrinkage of previously noted pancreatic head tumor with decreased angle of involvement, blah, blah, blah. But it says mild. What does mild mean? What's reduced mean? Where's the quantitation in that? To me, a radiology report is like an art critic describing a painting. That's about how useful it is to a buyer, right? So you got to like it, you got to not like it, I suppose, in that case. But if you could even quantify it, it would probably help, even in the art world. So what we really need to do is turn it into weather reports. Our weather reports are really, really, really quantitative, right? They used to be qualitative 50 years ago, right? Cloud in the afternoon, clearing tonight. Well, how much cloud? How much rain, right? How much sunshine? Well, we have all that information. We need to do it. We need to go in that direction. So I started a company, about 8 years ago, with 3 other, really, really experienced medical device software folks, including one of the founders of Geometrics. And the problem was to solve is to combine quantitative imaging with other medical data. In other words, take in EMR data and mine it quantitatively. The solution was then auto-contouring of lesions and create radiomic features along with other medical records from the EMR. The IP was software, obviously. But the trouble was the customers. Radiologists are not paid for radiomics. So really the customers are therapeutic customers. Our medical oncologists, radiation oncologists, and pharma companies. We had some pretty good investors, including Ascension, which is a very large medical device and associated with the Catholic hospitals in the United States. But we pivoted 3 times to try to find someone who wanted it. Radiologists aren't interested in it. They're not. They don't care. They don't care. Even though they see the images first, they would just want to stay reading their images. And if they don't get paid for it, unless there's a billing code, that they can say, oh, we're doing quantitative imaging now. How important is that? We'll get paid for it too. So the company couldn't raise investment this spring, and it was sold for very, very little to one of the largest drug companies who appreciate it. Genentech Roche bought it for a song because we couldn't get an investor. So in fact, one of the things is it did, semi-automated contouring heading towards fully automated GTV contouring to find the tumor in this example, and then making 2,300 radiomic features, all of which have been described in the literature. Nothing that hadn't been described in the literature was captured. So this was really the first and largest radiomics company, and it effectively went bankrupt because there was really no interest in radiology. So there's some lessons learned. Companies are needed to get medical products into clinical use. Disruptive technology is usually pioneered by startup companies. Incubation in the clinic and academic technology transfer groups are very important for success. Try to ensure that the investors do not have too much control over the company, like ADAC. Superseding significantly the technology of another company with better medicine, lower cost, easier use, and better throughput are the key. So those are the factors: better, lower cost, easier, and better throughput. If you can do all four, you're going to win. If you do 3 out of 4, you probably win. If you do 2 out of 4, you probably won't win. Customers' opinions are important, but they might not be helpful in the design of disruptive technologies, or even realize there's a need. Einstein once said that if he had an hour to think, he'd spend 55 minutes on the problem and 5 minutes on the solution. Special relativity wasn't a problem to anybody but him, right? There's a need to figure out how the technology will be paid for. If you can't find a way to get it paid for, it ain't going to work. It doesn't matter how good the idea is. And startup companies can enable research impact to be greatly multiplied, especially for applied sciences like medicine. So in summary then, applied scientists should consider the value of a startup company. Radiotherapy continues to be disrupted. Targeted therapy is in its infancy and will disrupt medicine. And I think mining data will eventually strike gold if you're a diligent enough prospector and there's a way to pay for your shovels. All right, thank you very much.
Thank you for this very interesting talk with a lot of examples. So I'm sure that we have some questions.
I just want to ask a question. Who here has started a company? There's 1, 2. Who? Come on, get hands up. 1, 2, 3. That's it? Okay. How many would consider starting a company if that was the only way to get their technology out? Okay. All right, good.
There are some.
Yeah.
Okay, and how many would have a question?
Thanks for your informative presentation you did and for these examples. But based for your experience, I am curious about some things maybe faced for anyone who would like to join the business. Since when you start a new technology for this, there is some challenges they face, like if this normal people, they will accept your technology as a patient for you, especially if there is something completely new for them, and especially if, when they hear about something about nuclear, so scared. So did you face this situation about your life and what you were doing just to make the normal people to understand the concept for radiotherapy or particle therapy? Just I want to, the second part, to give you the mic, or for the other one, how you make your technology available based for development countries related for the cost, in order for when we talk about some countries which is they need this technology just to make it available, especially when we talk about particle therapy. Unfortunately, we don't have until now about a clinical accelerator which is make cheap for just for protons because almost the country use the particle therapy, especially what I know they use for research, and this project is difficult for any country can do this project. How we can make this technology it's available for who's interesting just for clinical. Thanks.
Yeah, so let me talk about the last one first. So yeah, no, clearly Leo is starting in proton radiation therapy because the need is the greatest, the economic need, and the accessibility problem is acute, right? So Canada, my home country, doesn't have any proton machines because they're so expensive. There's many other countries around the world that don't have them as well. So not having to build a gantry and a giant shielded room, and often away from another center because there's no land near the center to build it, and involves new construction. And then it requires radiology, examining rooms, conference rooms, offices. So that just multiplies the cost. So if you can put, and I think we'll be able to do it, put protons into Linac vaults very soon, then that will make the accessibility of protons really interesting. You can put a small cyclotron into and an upright positioner into conventional vaults. That I think is in the horizon. So that will help a lot. And then in terms of people wanting to try new things, there's a so-called adoption curve, right? So the early adopters are the ones keen to try something. So thank God for them, right? They're the prophets. And if without them, you wouldn't have anything new in any field, right? So the early adopters always want to try something new. And then you eventually have this curve where you're starting to get a lot more interest, right? And then at the very end, it's the really super skeptics are the last people to buy, right? Or the last person to buy the old technology. I'd like to find out who bought the last simulator in the world. That would be interesting. Or who's going to buy the last proton center that has 3 gantries? That would be an interesting. Who's going to do that? Who's going to be marked as the last person to change? Well, I mean, that's what happens with disruption, right? You're sort of embarrassed with what you have. And in the case of protons, it's embarrassment that could last you 20 or 30 years.
Okay, we have another question here.
Hello, thank you very much. I found it interesting about your comments on B and CT. So it seems that one of the innovations from the companies are the accelerators to produce the neutrons, but I guess the boron is the key. So right. Are these the 10 times the number of boron? Are these used clinically now, or is it coming? And
well, they're not even in clinical trials, but people have made them and put them into mice and so on. So they're called carbaranes, and it's a carbon cage, and you can put 10 or 12 this onto a structure that's kind of like a soccer ball, and that can be attached onto targeted agents. Yeah, it does change. It probably does change the biodistribution of the targeted agent. So you might have to add an imaging agent that's lighter on top of it, instead of not having a carborane ball sphere on it. But the biodistribution will be whatever it is, and I still think we'll be able to find either a fluorophore for the same targeted agent in surgery, or you'll be able to put on a radioisotope as well as a partner to that for imaging.
Do you have a good estimate of when we'll
the
see
time?
this?
Well, I mean, things take minimum 7 years to get through. There's several papers describing these compounds now, so some of them will start going through the approval process, but I don't believe any of them have yet.
Thank you.
Okay,
Thank you for the excellent talk. I really learned a lot. I have a question because we see a lot of very famous professors. We also see a lot of very successful company, industry owner. How can you handle these two things kind of in parallel and in a very successful way? And what's actually your, let's say, suggestion for the young people for this direction?
Well, I wasn't always an entrepreneur. I was only a professor. but I really did get forced into it twice in a row. One because a great technology wouldn't have happened in a software, and then a great technology hardware. So I was a socialist when I moved from Canada to the United States, and we tried to give away the software, but the FDA, in fact, six clinics came to get the software for free, and take it home, and the FDA said they couldn't use it. So if the world were a better place, you wouldn't need a company, but unfortunately you do. And if you're a really, really good professor and you're patient and maybe go get an MBA to help or go work for a company for a while, like yourself, working for Varian for a while and a very good professor, you're the type of person that would be a good entrepreneur too. Because you've got to be a good, ideally, a good professor can become a good entrepreneur if they also learn something about business.
Okay, we have another question.
Thank you for your nice talk. I wanted to talk about point 3 directly behind you, and maybe if I understood you correctly, about targeted therapy and its use in parallel to radiotherapy. I like a good, nice conformal distribution as much as the next guy. But looking at nuclear medicine and their distributions, they're far, far away from that, and they're still effective. How would you say, is it a challenge that still has to be solved to reconcile these two, not advances?
You mean external beam and radiopharmaceuticals? Well, external beams are really, really great for big tumors. And the big tumors tend to have high pressures in them that prevent the diffusion of drugs into them. So they're a real problem. So I think we'll still have external beam to treat tumors that are billions of cells in some tumors, right? But if you have isolated tumorlets, hundreds of cells here and there, you'll never be able to image them, but they're there. And the targeted agents, if they as they get better and better, as the contrast to background, as I like to call, the amount of isotope in the tumor versus the amount of isotope in the blood is, if it gets to be 15 to 1, it's for sure going to work pretty well for those isolated tumorlets. And so that's why I think they should be used concomitantly for those patients that we pretty much sure that they already have metastases running around in them. Why should we wait to image them, right? Because if we wait to image somebody to see their tumorlets, they might have 1 million cells. It's hard to treat them. But if you can use targeted therapies and you don't have any pressure gradients for the agents to fight against, that's the time. That's the best time. The best time to treat cancer is when you can't see it.
Thank you.
Okay.
Yeah, thanks. I wanted to have your comment on a striking difference I saw. You did multiple startups, and they all had external funding, right? You always went to some, or like most.
Well, some I funded myself.
So you were the extended funding.
No, later on.
But
well, actually, I put $400 into geometrics. So.
Great investment. But on Sunday we saw actually that Cosylab, I think, is fully bootstrapped. And so could you comment on why you particularly went for always external funding?
Oh, I think we had to. There's no way we could have bootstrapped it, right? We needed to go get FDA approval. And that process, I estimated it would cost us $1 million to get FDA approval. And to put in, to make it a general purpose RTP. I was completely wrong. It was, it took pi times that. It took $3 million. So an academic planning the cost of something, you should always multiply by pi. Because you're not going to go straight across, you're going to go all the way around,
Okay, thanks.
So maybe building off the previous question about how you connect good professor and a good entrepreneur, I think one thing that's in common is you have to know which direction to go. So you have to know at what time you continue, like when you start a company, and what time you close it down or pivot. Mm-hmm. So what do you think? Is that a gut feeling, or is that the experience, or is there a strategy that what's the right?
Well, you got to fight like hell. You got to fight like hell not to get shut down, and you got to fight like hell to find a pivot. if you weren't successful. Yeah, I mean, you have to be really, really stubborn. I mean, Shine's a good example. You know, Shine was a university project at mortgage for a while. And they ran up $6 or $7 million in debt privately when they left mortgage very quickly because they didn't have the funding. And so I thought they were going to go out of business. But they just fought on, man. They went to the creditors and got them to forbearance. Because a creditor can force bankruptcy, right? So you know, if you want to be an entrepreneur, you better be willing to fight.
So, but how did you know when to fight? What was your instinct?
Well, my instinct's always to fight, right? And even in HealthMine, I was fighting to the last day to try to get that funded to outbid Roche. Because I'd done that once, right? So we bought an Australian company out of bankruptcy and it turned it into Leo Cancer Care. It was founded by Paul Keel, who many of you might know. And Paul, you know, had great ideas, but the manager, his business partner, wasn't very good and couldn't get funding. So it went into receivership. I bought out of the receivership. I'd been hoping to do that with HealthMine, as you know.
Okay, we have the last question here.
Hi. So you've talked about several disruptions in the field. I am just curious. Are you aware of any disruptions in the field of brachytherapy? Is it already dying, or is it being absorbed by another field?
No, I don't think so. I think actually actuarially, if you go and look at the local control and prostate radiation therapy, the best is both a combination of external beam and brachytherapy. It actually gives you the highest, you know, essentially, well, it's fractions of a % difference maybe, because it's already so well controlled with external beam. But adding brachytherapy does help. I think there are isotopes that you can shield better. So lower energy isotopes that you can put shielding so that you can actually use rotation of the direction of the isotope, either, you know, shield half of it or shield three-quarters of it. That could give you more flexibility in planning so that you could give higher doses to the target volume and protect normal tissue. There's nothing, brachytherapy is pretty damn conformal and can be even improved. But it's still messy, right? Still invasive.
Okay, so thank you for this wonderful talk and all this interesting discussion that followed. It was nice to have you here.
Thank you.